Low-density lipoprotein receptor-related protein-associated protein 1, also known as LRPAP1 or RAP, is a chaperone protein which in humans is encoded by the LRPAP1 gene. LRPAP1 is involved with trafficking of certain members of the LDL receptor family including LRP1 and LRP2. It is a glycoprotein that binds to the alpha-2 macroglobulin receptor, as well as to other members of the low-density lipoprotein receptor family. It acts to inhibit the binding of all known ligands for these receptors, and may prevent receptor aggregation and degradation in the endoplasmic reticulum, thereby acting as a molecular chaperone. It may be under the regulatory control of calmodulin, since it is able to bind calmodulin and be phosphorylated by calmodulin-dependent kinase 2. Ldl-Receptor-Related protein-associated protein has been shown to interact with LRP2. Lipid metabolizing proteins may elevate susceptibility to dementia, leading to differences in genetic makeup. PCR restriction fragment length polymorphism technique is used for genotyping of LRPAP1 intron 5 insertion/deletion. The studies suggested that DD genotype and *D allele of LRPAP gene showed increased frequency for degenerative dementias on comparison with the control group, and that LRPAP1D allele remarkably increases the vulnerability to degenerative dementias. On genotyping of LRPAP1 polymorphism is observed because of 37 base pair insertion in intron 5. Also, insertion allele being larger than deletion allele makes possible in detecting difference by gel electrophoresis. Suppression of receptor binding domain of LRP-LDLR is due to overexpression of LRPAP, the protein product of LRPAP gene. LRP gives protection across LDL by LRPAP, and its downregulation may be subjected for an elevation of LDL and a related neuronal toxicity, as LRP supports in binding of ligand and internalization of LRP ligands like ApoE-enriched LDL cholesterol and a protein. Insertion/deletion is an intronic polymorphism of LRPAP gene, influencing DD genotype and D allele for the synthesis of LRPAP protein can be LRP-mediated mechanism contributing to dementia. Concern for developing sensitivity for dementia is because of several shared common genetic platforms, and DD genotype or D allele of LRPAP gene may be one such. So on 37BP insertion/deletion that was studied as an intronic polymorphism, it could be having an unintended pursuit for lipid receptor protein by regulation of LRPAP expression, or it could be in linkage disequilibrium in addition to other biologically relevant polymorphism in the LRPAP1 or an adjacent gene in chromosome 4. Results being consistent with earlier study where the authors have endowed deletion allele frequency clearly high in late-onset Alzheimer's disease patients on comparison with non-demented aged controls. Mendelian forms of myopia has been identified in 4 consanguineous families and are the likely causal mutations implementing exome/autozygome investigated to recognize homozygous truncating variants in LRPAP1. Influencing TGF-beta activity. Chaperone of LRP1 is encoded by LRPAP1. Notably, salient deficiency of LRP1 and upregulation of TGF-beta in affected individual cells, the GNOME data being consistent on the importance of TGF-beta in remodeling for the sclera of myopia and the increased frequency in individuals for myopia having Marfan syndrome, which has characteristics of upregulated TGF-beta signaling. Analyzing the absence of the normal protein was done with immunoblot for affected individuals having LRPAP1 mutations, revealing the mutations in LRPAP1 probability of loss-of-function mutations. Encoding: low-density lipoprotein receptor-related protein-associated protein 1, LRPAP is a largely expressed gene, and a 357 amino acid protein thought as a chaperone binding and protecting the lipoproteins receptor-related proteins LRP1 and LRP2. A model suggested by a study wherein LRPAP1 leading to deficiency of LRP1, which was responsible to perturbation of TGF-beta regulation and might cause abnormal ECM remodeling in the eye development. On observation for increasing axial length was one of the salient features of Marfan syndrome, also resulting in TGF-beta supported the model. Therefore, individuals having myopia responding to therapeutic strategy initiated before ECM remodeling could be considered as an approach for individuals with LRPAP1-related myopia.